Hello everyone, we are back. I hope that you can not see me too much, but at least hear me for the best of you. And so now after this opening session, we start by a first keynote. We have 3 fantastic keynotes during these 2 days of the virtual ETPN 2020 event. And it's really a great pleasure to leave the floor first to Ruth, because Ruth Schmidt will introduce Patrick Baumhoff, our first speaker, and then I will show you the lecture of Patrick, and he is live with us, as you know, to answer all your questions. Please don't be shy during the keynote. Ask as many questions and comments as you wish. We have time after the session, before the lunch, to answer your questions. I mean, Patrick will. Ruth, the floor is yours.
Thank you very much. It is a great pleasure for me to introduce my good friend, Patrick Baumhoff, as the first keynote speaker. Patrick is Vice President Formulation and Delivery at CureVac. He got his training in chemistry at the University of Leipzig, and his scientific expertise includes chemistry, pharmaceutical sciences, and immunology. He joined CureVac in July 2007 when he was responsible for the development and preclinical testing of new formulations for mRNA vaccines and therapeutics. He's the inventor of several patents, and he co-authored several publications on mRNA technology. Currently, he is heading the Department for Product Design and Formulations, and he is program manager for the Rna Optimizer program. And he's also an active member in the Controlled Release Society. That's where I got to know him. I'm looking very much forward now to his talk, mRNA-based vaccines. Please, the floor is Patrick's now.
Hello everybody, my name is Patrick Baumhoff from CureVac AG, and today I will show you an antibody as a therapeutic option for the European technology platform nanomedicine. Since CureVac is a publicly listed company, I have to show you my favorite slide, which are the forward-looking statements, and you should not read through this. And after showing this to you, I will dive, hopefully, in an adventurous journey with all of you for the next 40 minutes on messenger RNA-based therapeutics. This is a revolutionary basic principle to use messenger RNA as a data carrier for healthy messages. As you all know, the source code of life is safely stored on DNA in the cell nucleus of all living cells. The information that encodes principally all of the proteins that your cells utilize is encoded in nucleic acid code. And in order to translate a protein to produce it, the information has to be transferred from the cell nucleus to the ribosomes in the cytosol, and this is achieved by transcription of the information from the DNA into a messenger RNA that is transferred to the cytosol and then is translated into the proteins via the ribosomes. And we at CureVac are using this as a natural messenger for health. So you can principally use an externally generated messenger RNA to give the body the information that he requires to generate its own therapeutic protein. All what the cell needs principally is the right information. The basis of all of this is that you need an engineered messenger RNA, and it's also the basis for all the CureVac's technology platforms. Because if you start with a sequence that you derive out of a nucleic acid sequence database, you will end up with a messenger RNA that performs very poorly. You can see that you have a very low peak expression and also a very transient decline of the expression because of the short half-life of your messenger RNA. But there are tools and also rules that you can follow to render and change the fate of the messenger RNA inside of the cell cytosol to keep it within the ribosomes and to drive a really high and a long-lasting protein expression. And with such a powerful messenger RNA sequence, you are really able to now think about using this for delivering therapeutic information to the cell and generate medicine. Therefore, messenger RNA holds an unlimited opportunity. So depending on the protein that you express, you can prevent or treat diseases in the infectious disease area. You can use it for prophylactic vaccines in global infectious disease settings. You can use it against cancer for immunotherapy, and you can also use it to replace proteins, endogenous proteins for molecular therapy. And here you can think of proteins like insulin, ion channels, defective enzymes, or antibodies. And therefore, the potential to record healthy messages for different indications on the data carrier mRNA is limitless. Today in my talk, I will concentrate on antibodies as a therapeutic option. So if we talk about messenger RNA encoded antibodies, we use the body as a bioreactor for producing the antibody in situ, directly in your body. And depending on the, your target molecule, if it's a protein, if it's a toxin, some tumor cells, tumor antigens on tumor cells, bacterial pathogens or viral pathogens, you identify your target molecule and the antibody modality that you want to use, and you then transfer this information into your messenger RNA, put this messenger RNA into some kind of delivery vehicle and transfect in situ your cells that then will produce the functional protein. And depending on your antibody-mediated effect, you can use it for target inactivation just by binding, for example, toxins, for opsonization to impair pathogens, or even for target killing if you think about of CAR receptors. And what messenger RNA also holds as a potential in addition is that you also can think about using intrabodies, so intracellular antibodies. That is something that is not as good possible with a recombinant protein, but with messenger RNA that is produced in the cell cytosol, the cytosolic expression of intrabodies is clearly possible. So when you are thinking about producing or designing therapeutics, it's clear that you need a lot of experience and to optimize in order to come to a functional and also suitable therapeutic in the end. And CureVac has invested over 20 years into this optimization experience, and we came up with a process that we call Rna Optimizer, and that is optimizing the messenger RNA therapeutic on several different levels. The first level is protein optimization, so the selection of your target protein sequence. And since we have a genetic concept, it's for us rather easy to change properties of the protein sequence very easily just by exchanging codons. And here we can, for example, think about altering the cell localization, bringing something to the cell nucleus or to the cell cytosol or secrete it. We can add protein domains to modify protein properties like half-life prolongation. We could tune and find the optimal signal peptide for a secreted protein for your target cell that in the end produces your protein. And once you have done all of this, you have principally identified or defined your final protein sequence of your therapeutic. Then you go into a process for messenger RNA optimization. And here you have several levels where you can optimize your messenger RNA sequence. First of all, you can think about open reading frame sequence optimization. So usually, since the genetic code of the amino acids, it's degenerated, where you have several codons coding for the same amino acid, you can change the open reading frame sequence by exchanging codons without exchanging the amino acid sequence. And thereby you can enhance, for example, GC content. You can think of removing or introducing three-dimensional structures, hairpins, and other structural elements. And all of this can principally influence the potency and the half-life of your messenger RNA. But there are also untranslated regions that are not encoded to the protein, and these untranslated regions are very important for the fate of the messenger RNA inside of cells. So once inside of the cytosol, the messenger RNA is recruited to the ribosomes and it has to stay in there. Otherwise, the RNA will be easily degraded also within the cytosol. So the UTRs principally fine-tune the half-life of the messenger RNA inside of the cell. Once you have then optimized your messenger RNA sequence, you principally have defined your drug substance. So your untranslated region sequences, your open reading frame sequence, and thereby the RNA drug substance. Then, depending on your target application, where you or your bioreactor inside of the body, you will then need a nanoparticular formulation. And this clearly will be different if you want to use the eye as a bioreactor or treat diseases in the eye. If you want to use the muscle as a bioreactor or a vaccination site to elicit an immune response, or use the liver as a bioreactor to produce high levels of protein from the messenger RNA. And thereby, formulation and delivery is an important step for all messenger RNA-based therapeutics. In the next step, I want to shed a little bit more light into the definition of the messenger RNA sequence. We at CureVac were really trying to understand the origin of the regulatory sequences of a messenger RNA-based sequence. And for being able to do this, we principally screened different cell samples, different cells like hepatocytes, muscle cells, nerve cells, and different conditions like homeostatic cells or stressed cells. And then we tried to identify sequences, messenger RNA sequences that were very stable or very high expressing. You can do this by a method that's called ribosome footprinting, where you principally fish the messenger RNAs based on the amount of ribosomes that are on the messenger RNA itself. And usually a messenger RNA that has more ribosomes on it is highly expressed and usually also longer stable. And with pulse chase experiments, we were also able to define, to get an insight into the half-life of these messenger RNA sequences. And we screened over millions of different sequences, natural sequences used by your own cells. And what we came up with were different sequence elements called 5' UTRs and 3' UTRs that we ranked in these assays for potency and also half-life stabilization of messenger RNA. And the interesting thing was when we were using these UTRs in different contexts, for example, looking into a liver hepatocyte, where you should look here at the left side panel, you can see that the combination of different 5' UTRs and 3' UTRs lead to different potency in terms of ribosome footprinting of the different sequence combinations. And you can see that the combination of this 5' UTR and that 3' UTR is the most potent in hepatocytes. When we then switched to a different differentiated cell type, for example, muscle cells, the picture changed a little bit. It changed not completely, but in details. And these details are very important if you strive to find the optimum sequence for a different purpose. And here you can see that in this case, the combination of the most potent 3' to 5' combination changed. And we utilize this, and if you have such a problem that you have a library of different sequences that principally are differently potent in a different context, you usually want to do a screening. And this was the reason why we started at CureVac to invest into high throughput synthesis of messenger RNA. And what we did here is that we generated a process that enables us to generate libraries of messenger RNA sequences. And here you start principally with the gene of interest. For example, if you're looking for an optimal sequence expressing erythropoietin, EPO, or for example, the S1 antigen of SARS-CoV-2. You start with the gene of interest, and then we have a plasmid DNA library that principally confers already the pre-selected 5' regulatory sequences and 3' regulatory sequences that we identified over the past 20 years as the most potent ones. And you clone in your gene of interest and generate a messenger RNA library out of this that then can be directly transferred into cells. And you can screen in your particular cell type of interest, for example, hepatocytes, for the most potent sequence within this cell line and with this gene of interest. And thereby, you have a very efficient and very smooth process to down select the most relevant optimal sequence. Here you can see an example how this would look like. Here we used an on-cell Western blot on 96 well-plated cells and can directly read out the amount of protein that was produced from the different messenger RNA sequences. And you can clearly see that there are very different expression levels throughout the plate, but there are several hits, several sequences that perform on a very high level. And in the next step, you can then screen the most well-performing messenger RNAs directly in vivo for your best candidate. I want to give you an example here. What we did, for example, for the Cas9 enzyme. Here we started with an UTR combination that is usually performing very well in several different contexts. And we used here this and compared this messenger RNA against a commercial modified messenger RNA that you can acquire just by ordering through an CMO. And what you can see is that the standard RNA from CureVac was roughly 6-fold better in expression in vitro compared to the commercial standard. We then also tested the knockdown of C3 serum levels utilizing this Cas9 coding messenger RNA together with a guide RNA complexed in a lipid nanoparticle given to liver of mice in vivo. And you can see that you had a very efficient knockdown of the C3 serum levels. In a way, you can now say clearly that could be a product, and we can directly move to the pivotal preclinical trials and also move to the clinic. But here we wanted to also know if we were already at the best stage of messenger RNA sequence optimization. And therefore, we conducted our screening assay for Cas9 in liver hepatocytes. And what you can see is that from this library of 83 different messenger RNA sequences, the red one here, number 10, was the sequence that we used in the slide before in these experiments. And you can see that we were able to increase the expression in liver hepatocytes by a factor of 5. You might not think that's a lot of effort for a factor of 5, but if you consider you would bring down the dose of an intravenous infusion from 1 mg per kg to 0.2 mg per kg, that's a very significant impact in terms of cost of goods, tolerability, and also efficacy. As you see, it's very important to optimize your messenger RNA sequence. Now I will switch gears a little bit and we will dive into using the liver as a bioreactor for RNA antibody expression. Why would we use the liver as a bioreactor for RNA antibody? So it's an interesting target for several different reasons. First of all, the liver is a tolerogenic organ. So it's an immune privileged organ. And I think that's also the reason why there are several viral infections that are persisting in the liver, like HBC. And therefore, in this case, we use this Achilles heel of the liver in our favor. It's also the largest organ and it's well vascularized, so it's highly accessible. And for us, it's very important to reach and transfect as much cells as possible, because the amount of producer cells maximizes the RNA antibody output. And in principle, we usually see a very linear equation between transfected cells and protein output. And third of all, there are established delivery systems for efficient delivery to liver hepatocytes that were known from the siRNA field, and we principally built on this experience and use this lipid nanoparticle-based technology in order to reach liver hepatocytes to use them as a bioreactor. So to express an antibody via messenger RNA, you have to express two different protein chains. So one chain is the heavy chain of the antibody and the other is the light chain of the antibody. And therefore you have to generate two different messenger RNA. One encodes for the heavy chain of the antibody with the respective signal peptide, 5' UTRs, cap, and poly-A tail, and a messenger RNA sequence that encodes for the light chain. We usually mix these two messenger RNAs and then generate the lipid nanoparticles out of this. And if you principally transfect this into cells, in this case, I think it was BHK cells, you can see that you can see both the light chain and the heavy chain in the lysates of the cells, but you also can find both of these in the supernatant of the cells. In order to do this also in vivo, to load liver hepatocytes with our messenger RNAs coding for the heavy and the light chain, we used a lipid nanoparticle composition that we are acquiring by a collaboration with Therapeutics. Usually these lipid nanoparticles consist of an ionizable aminolipid, which is the key component that drives the efficacy and tolerability. Usually a PEG lipid that stabilizes the particle against agglomeration, aggregation, and keeps it stable during shelf life. And structural lipids, which are usually cholesterol and or DSPC or DSPE, that principally form the lipid nanoparticle, give them flexibility and also shape. Usually this is generated by an ethanol dissolution process where you have the messenger RNA mixture in water, usually at a low pH, and you mix this with the lipid components that are solved in ethanol in a rapid mixing process. Could be in TPs or could be in a microfluidic system. And in the end, you would end up with spherical nanoparticles that have the PEG lipid in the shell and the messenger RNA in the interior. And if you inject these particles intravenous into mice, you usually get a very efficient uptake in almost all hepatocytes of these animals. And here you can see immunohistochemistry staining for the messenger RNA, and you can see that all of the liver hepatocytes are positive for the messenger RNA, whereas no change in the histology of the mouse liver was seen at this particular dose. We then moved into the animals with messenger RNA encoding for 2 different antibodies that are known. One was an anti-rabies antibody, so an antibody that recognizes an antigen of the rabies virus, and one anti-influenza B antibody. And what you can see is that we get a very linear dose-response curve in terms of antibody titers that are measured in the sera of the animals when we increase the dose of the injected messenger RNA. And you can see that this dose-response curve is very similar for both of the antibodies. And you can see in this case we reached levels between, depending on dose, 1 microgram up to 10 microgram per ml. We were then also interested in the functionality of the antibodies expressed by the messenger RNA in situ in the mouse, and therefore we utilized for the anti-rabies the standard VNT assay that is recognized by WHO for as a correlate of protection. And this is conducted by standard laboratories, and we send the samples of the mouse sera to these laboratories, and they measure the VNT. And you can see that we get an also nice linear increase in virus neutralization when we increased the messenger RNA dose, similar to the amount of antibody that we were measuring in the blood. And you also can see that at a low dose of 2.5 microgram, which is below 0.5 mg per kg, the mice were already be protected against the virus. In the next step, we were also interested in the in vivo kinetics of the messenger RNA encoded IgGs. And as you know, the usually half-life of an IgG is roughly 21 to 28 days. And here you can see that we had a peak expression at day 1, but very stable serum levels for over 3 weeks, which is very similar to the recombinant antibody. In the next step, we went into a lethal virus challenge model, and we had 2 different modalities how we tested the messenger RNA for functionality. So the whole concept, and the first setting was that we had a prophylactic passive immunization of the mice, where we treated the mice 24 hours before viral challenge with the messenger RNA encoding for the anti-rabies antibody, and giving the body the possibility to produce the antibody and have the antibody already circulating in the animal when the virus was challenged. In a different setting, which is more a realistic scenario, we challenged first the animals, and 2 hours after virus challenge, we gave the antidote, principally the messenger RNA encoding for the anti-rabies virus antibody, and then we're following survival. And what you can see on the right-hand panel, that in both cases, in the anti-rabies mRNA therapeutic setting and also in the prophylactics setting, all of the animals survived the challenge. Whereas the ones that we treated with the anti-influenza B antibody that is not protecting against the rabies virus, the animal succumbed against the lethal challenge. We were then transferring this concept also to non-human primates already back in 2017. And what you can see here is also there, we get a very similar PK compared to a recombinant antibody in the phase 24 hours and after. Clearly before, usually with an antibody you inject at very high doses, so you enter the therapeutic window from above, Whereas for messenger RNA, we usually enter the therapeutic window from below because we start at 0 and then we have the ongoing expression that then leads to an increasing PK of your antibody in this case. And you can see with the technology that we had in hand in 2017, we reached levels roughly around 1 microgram per mil in these animals. And we were continuously investing in our technology, and we're optimizing the lipid nanoparticle, but also the messenger RNA sequence. And over the past 2 years, we were able to principally move the borders, and now we reach levels in the animals of roughly 30 to 40 micrograms per mil at the same 1 mg per kg dose. And such antibody levels are really relevant and can be used for therapeutic interventions also in larger animals and ultimately in humans. We were then also looking into different messenger RNA encoded antibody formats. One of very popular format is the Camelot single domain antibodies, VHH. And if you fuse multiple of these VHHs together, you come to an VNA, an nitrolysing VHH-based nitrolysing agent that was developed principally by Chuck Shoemaker. And we were using such sequences and were encoding them on messenger RNA. What you can see here, they had a signal peptide, then the 2 domains and an albumin binding peptide for enhancing serum stability of these Camelot antibodies. And you can see that we were using here in Zika toxin specific VNA, that we were getting very high levels of the protein in the supernatant of transfected cells and also in animals. In animals, we tested also 2 different types of VHH. One was against anti-botulinum toxin VNA, and the other one was against Zika toxin. And you can see that we reached very high levels for the botulinum toxin VNA for several hundreds of micrograms per mil. For the Zika toxin, we reached a little bit lower levels, but also very significant levels in these animals. In the next step, we were interested if we can principally also raise a therapeutic relevant effect with these VNAs encoded by messenger RNA. And again, in all of these cases, the messenger RNA was encapsulated in lipid nanoparticles and transferred intravenously into liver hepatocytes to use them as a bioreactor that produces our proteins directly in situ. In the next step, we challenged mice 6 hours post the toxin challenge, and in this case we used botulinum toxin. That is a very stringent and harsh assay principally, because the mice succumb very, very rapidly due to the botulinum toxin effect. And you can see that even 6 hours post challenge, the messenger RNA was fast enough to principally prevent the toxic effect of the botulinum toxin in a similar manner compared to the recombinant botulinum VNA. Excuse me. Whereas the animals that were treated with the Zika toxin specific VNA succumbed very fast. As a next step, we were also looking into if we reach relevant levels and functionality for antibodies that can be used in oncological settings. For this, we used a mouse model that utilized Ragiluk expressing tumor cells. Ragiluk is very nice reporter cell line. It's a very rapid going tumor, and you can easily follow the tumor growth by measuring the luciferase in these mice, luciferase expression in these mice. And what we did then after the tumor challenge, we treated the animals with our antibody coding for rituximab, again using the liver as a bioreactor, and compared this to the recombinant rituximab protein, and followed the tumor growth and survival of these animals. And on the left-hand panel, you can see the luciferase signals evolving over time, and you can see in the control group, untreated group, that there is a rapid growth of the tumor cells, which you can follow by the steep rise in luciferase signal. You can see that you get a similar fast growing of the luciferase signal when you use a lipid nanoparticle expressing for a control antibody RNA. And you get an inhibition of tumor growth if you use the recombinant rituximab in 200 microgram recombinant dose. And this was given 2 times a week, as also was the messenger RNA. And you can see with the rituximab encoded messenger RNA encoded rituximab, we were seeing a tight control of the tumor cell growth. And this was also reflected then in a significant difference in the tumor survival rates. In the next step, we were then going a further step. And what is also very interesting in the field is currently the move to bispecific T-cell engagers or bispecific antibodies. Here we chose blinatumomab as an example and wanted to show that also these type of concepts can be easily transferred to a messenger RNA-based approach. So we used the sequence that was FDA approved for the bispecific antibody targeting CD3 T-cell receptor and the CD19 antigen on B cells. And what we did here, we used the bispecific antibody on antibody coding RNA, where we had fused the anti-CD19 and the anti-CD3 together, injected mice, and followed the tumor growth and survival. And what happens principally is that the bite principally engages the T cell to the tumor cell, and that facilitates then tumor cell killing. Here you can see we used a mouse model. It's a little bit complicated. It's a NALM tumor model. What you have to use here is a skip mouse because you have to supplement human PBMCs as a type of immune system that can be recognized by your blinatumomab. So it's a little bit artificial, but you can see that, that in the control, the tumor cells, the 9 tumor cells are growing very fast and principally growing rapidly out. And if you principally supplement an antibody together with an PBMC cocktail, you can control the tumor growth of these tumor cells. That principally was the data that we utilized with a lipid nanoparticle and using the liver as a bioreactor from antibody. Now I want to switch gears a little bit more and also want to look, since we have a messenger RNA-based concept, we could principally use every cell in your body to produce the antibody. And as you know, going to the liver via an IV infusion is quite invasive. And also you need high amounts of messenger RNA. We were also interested in using the muscle as a bioreactor for an antibody, which is much more accessible and much more suitable in scenarios where you have to vaccinate passively a lot of people. And as I said, muscle is also an interesting target for several reasons. It's less invasive administration compared to an IV infusion. It could enable direct cocktails by utilizing several injection sites. And the The downside for muscle is that you clearly have lower amount of producer cells. And as I said, this principally direct correlates with the amount of antibody that you will achieve with your messenger RNA-based therapeutic. And therefore, the muscle as a bioreactor might not be suited for every single an antibody concept, especially the ones where you need really high amounts of antibody in the blood. And the muscle as a bioreactor might not be suitable. But nevertheless, we were looking into this, and we had a project together with Arcturus Therapeutics for the lipid nanoparticle technology that we were using for the muscle as a bioreactor, and US Amrit, who had the expertise in the smallpox relevant animal models. And we were using here a passive immunization against smallpox as an example how an antibody could look like for an intramuscular administration route. To remind you a little bit about smallpox, there are protective antibodies known against smallpox. 3 antibodies are described that are able to principally inhibit or can be protective against smallpox. 2 of these antibodies, anti-B5 and anti-A33, they bind against the major viron on surface proteins. And 1 is the anti-L1 antibody that binds an antigen that is only present on the immature virons of the viral infection. And you might not know, but usually during a smallpox infection, you are exposed to both major and also immature virons, and both of them are contagious. We were then looking into the expression levels using the anti-L1 antibody as an example. And we formulated a messenger RNA and injected here principally rabbits as a model, because rabbits are also quite large animals, and they have a good muscle injection site. And we were interested in antibody levels in the sera. And you also have to know such a rabbit has 125 to 150 milliliters of blood volume. And we observed here that we were able to reach sera titers of up to 9 microgram per mil in the sera of these animals. And what we were also looking here into was a comparison of a single needle injection that is given on the chart in black against a single jet injection. So in jet injection is a needle-free administration method where principally via pressure you generate a liquid beam that punches through the skin. And what we've seen in the past is that this enables you to a better tissue distribution of your injected solution. And this principally means that you have a better distribution of your messenger RNA therapeutic within the muscle, reach more producer cells, and thereby getting higher expression levels. And this is what we also were observing here. So in principle, we reached roughly a 2-fold higher expression level when we switched from needle injection to single jet injection. What you also can see, if we distributed several injections over the different muscles in the animal, we were able to increase the observed sera levels in the blood. And this again shows you that there is a direct correlation between transfected cells and the amount of protein that you get in the end. We were then also looking into the functionality of the antibodies that were circulating in the sera of the animals. And the US Amrit conducted here a plaque reduction neutralization titer, and they compared it against the recombinant C7011 anti-L1 antibody at a 10 microgram per mil dose. And you can see that the different animals with different amount of antibody levels were principally utilizing a neutralization titer similar to the recombinant protein when they reached similar levels in the ELISA, in the COR. And what was also very nice that the day 3 interpolated value of the expressed C7d11 antibody in the serum was similar to what we observed in the neutralization assay. With this, I would come to an end of my talk, and I just wanted to shortly recap what I've put you through the journey over the last minutes. And I think I have shown you that our CureVac's research engine opens up a broad space for antibody-based product development. As I have shown you, antibody show therapeutic effects in several different disease models. I have shown you data on oncology, on antitoxins, on passive immunization, and I think all of these applications are really relevant corner pieces of human health. You also have to think about that messenger RNA is a very versatile production process. For all the messenger RNAs that I have shown you throughout my talk today, we utilized the same platform production process, the same equipment, the same facilities. If you think of a recombinant protein, that would not be possible to switch from one day to the other to a new protein target. We just switch the input plasmid DNA and we switch from an anti-rabies antibody to an rituximab encoding antibody. It's also a very facile transition from the R&D stage to the CMC stage. We also were showing that we can principally do all of the various antibody formats that are currently circulating in the literature and also in the different research labs that can be addressed by an antibody. So I have shown you full antibodies, single chain antibodies, we can express fat fragments, we can do bispecific antibodies, intrabodies, VNAs, Camelot antibodies. So in principle, every single application that you can think of is principally addressable by an antibody. And we are also having a lot of data for the application of an antibody in various different tissues. Today I have shown you the examples on liver and muscle, but we also have examples for direct tumor injection, for applications in the eye, and also applications in the lung. And with this, I hope that I have given you some insights into messenger RNA-based therapeutics, an antibody in special. And I hope that all of you had a nice time with me, and I'm really looking forward to your questions. Thank you.
And we are back. Can you hear me, Patrick?
Yes, I can hear you loud and clear. Can you hear me also?
Yes, you can turn on your video if you wish. So yes, perfect. We can see you and hear you. Well, first of all, thanks a lot. That was an amazing talk. I mean, I learned a lot on mRNA vaccines, and some of the data were just astonishing. So congrats for that. I will wait for some questions, but I do have some questions for you while the crowd is warming up. Well, you won't be surprised that some of them would be around the technology by itself, and maybe some of them would be just to have an update of what you can say about your program against COVID-19, indeed, which is the hottest topic that we could imagine, indeed. So first of all, let's talk about the development. You've shown us that it took years and years to improve. It's really a complex engineering process. And my first question is a bit naive, but in all around this journey of production of these complex objects, Was nano the nano part, the complex part, or finally it was just quite an easy carrier to engineer and to optimize for you? Was it the tough part, the nano part?
Yeah, the tough part in nanoparticles, I think, is mostly the analytics and really to pin down from a delivery standpoint. If you are using a nanoparticle, you really need to understand the key critical quality attributes that you have to follow, especially during upscaling when you go into larger production steps and you change maybe process steps. For usually you have at R&D production, you have dialysis as a buffer exchange method. Later on you have TFF. So there are a lot of things that will change, and it's usually hard then to really pin down if you changed the material and also the properties of your material. Sometimes just measuring a size is not enough. Yeah, and there it becomes complicated. That is the nanoparticle part. On the drug substance part, there are also some aspects, but I think here, especially on the translation of the production and making this really commercial, the analytics and better understanding of processes will be necessary.
Great, crystal clear. We have lots of questions for you, no big surprise. First one is from Stefano Salmaso from UniPD. He said, thank you for the nice talk. I was wondering how long are the antibody expressed, which correlate with the protection time of the therapy?
Yeah, usually you have to discriminate here between the half-life of a protein expression that is driven by the half-life of the messenger RNA inside of the cell, and then the half-life of your protein that you produced. Since the half-life of the antibody is usually in the range of 3 to 4 weeks, clearly the messenger RNA is only expressed via the first 1 to 3 days. And this messenger RNA expression is building up your sera level, and at some point the messenger RNA expression ceases principally, but your protein still remains with its usual half-life.
Very clear. Next question is from Sue Metcalf from Leaf Nano. And Sue is asking, is there an antiglobulin response to the therapeutic antibody first? And also, is there spread that might induce antibody response to liver or muscle?
Mm-hmm. Yeah, so anti-drug antibodies are an unusual phenomenon. Here you have to separate principally between an anti-drug antibody against your drug substance and drug product, that would be a messenger RNA and a nanoparticle at the one part, and the protein that you express on the other hand. And if we express, for example, in the rabbit model, we have seen occasionally anti-drug antibodies against the human chimpanzee antibody that we expressed. That is usually, it's very hard to study ADA with human proteins in animals. That is usually giving some background noise. You also have some immune stimulation by your treatment. You give a messenger RNA in a lipid droplet that is virus-like. So the immune system is searching for these things. And you have to master and engineer for this to not raise an immune response directly against your protein, that you vaccinate against your therapeutic protein. There are some rules and also a technology to do this. And here we are clearly looking very close into this. The data that I've shown you, in the most cases, we haven't seen such things. It also always depends on the immunogenicity of your protein. For example, the VNAs in mice, they had a very steep drop after 5 days, which is related to an immune response because they are foreign proteins in an animal.
Okay, very clear. Now we have two questions by Raymond Schifflers, our dear co-chair, vice chair of the TPN, professor of nanomedicine in UMC Utrecht. First question is, how did you approach the multitude of variables that together make up the critical quality attributes? And we all know in nanomedicine, it's part of the biggest challenge that we all have to face. And the second question, if I may, still from Ray, is, as you say, the mRNA expression is relatively short-lived. Are the best therapeutic approach depending on engaging an amplification step, like for instance, the immune system?
Mm-hmm. Yeah, first question was related to the critical quality attributes.
Absolutely.
Here, this is a huge effort that you can do with a defined product when you have something like currently we are looking into the COVID-19 vaccine that we are developing. Then you have to do a lot of analytics to understand impact of process changes on these analytical methods that you apply. And you then also check the biological activity and you try to find correlations that can guide you to really identify if you have made a significant deviation or a significant modification to your process that impacts then afterwards your efficacy.
Okay, and for the amplification step?
Yeah, the amplification step, it really depends on what type of therapeutic you want to do. If you compare messenger RNA to a DNA, for DNA, you usually have a long-term expression, but that is less predictive because in some cases you have integration in the cell nucleus and you have varying expression levels due to the different efficacy of the transfection of cells. With messenger RNA, we get a very predictive PK that is transient, clearly, but it's predictive. And for some applications, it might not be the best solution. You could change this by using, for example, self-amplifying RNA concepts. That comes usually also with some edges, also thorns, not edges, because you have then also these structural proteins that you required for the amplification step of the messenger RNA that can also be a raise immune response and ultimately lead to the depletion of your producer cells. Here, it's really a case by case, which way forward is the best one. For example, for vaccines, I believe that a normal messenger RNA that has a pharmacokinetic of antigen exposure over a few days, which is very similar to a viral infection, is favorable against a prolonged antigen exposure over several weeks.
Okay, great. We have time for 3 or 4 more questions before the lunch break. So the first one is from Rna Optimization. First, congratulating you for the nice quality of the talk, Patrick, indeed. And then asking, do you affect natural function of the body cells, for instance, liver cells, if you indeed such highly optimized expression of delivered mRNA to produce high amounts of proteins?
Yeah, you could think of that you might disturb cell homeostasis if you produce a lot of protein on top of the usual duty of the cell. I think the question goes into this direction. Is this correct?
Unfortunately, I cannot comment because the
question. Then I would take this as a given. We haven't seen a huge impact so far on the cells via the overexpression of certain proteins. This is only true as long as your overexpressed proteins does not have knock-on effects. For example, we know for some proteins, they at some point lead into toxic effects. If they are overexpressed, you can then disturb cell homeostasis that can have a detrimental effect. But usually you can balance this by the messenger RNA doses that you give in order to steer the protein levels. Most relevant, we have only seen this in most cases for intracellular proteins. If you have secreted proteins, usually there is no negative impact.
Great. Last question from the crowd, and then I will have a final one if you're okay. So the question is from Dwigu Yilmaz. I hope I pronounce it almost correctly. Is there a size limit for the mRNA target for it to express efficiently, or is the size only important for the efficient encapsulation into the nanocarrier system? Thanks.
We have worked over the past 20 years with messenger RNA constructs ranging from 45 kilobases up to over 10,000. The most impact that you have is if you go into extreme sizes that production gets a lot more difficult. And the messenger RNA at some point also gets susceptible to shear stress that is impacting yields and generating side products that you do not want to have. In terms of encapsulation, for example, in lipid nanoparticle, we were not seeing a lot of impact. For example, Cas9 is a quite big messenger RNA. I think it's 9,000 bases compared to, for example, an viral antigen that's usually between 2,000 to 3,000 bases. Here we were seeing a slight increase in nanoparticle size. It's clear that the amount of messenger RNA molecules per single lipid nanoparticle will be lower because you package usually via microgram of RNA versus micrograms of lipids. And your molar ratio then is clearly different if you have a 10-fold larger messenger RNA. What we also see then that the dose scaling, if you do it on a mg per kg, for example, your real dose in terms of strands is lower by the margin of larger construct that you have that has some impact.
Indeed. And now to finish, you won't be surprised on my last couple of questions. So the first one is indeed about having, let's say, an update about what you can say about the level of preparation of your solution against COVID-19 first, because this is exactly what we were saying in the introduction session, like nanomedicine has something else to bring. So if you can give us the level of preparation, level of development, any clues on any, let's say, insights on the comparison with recombinant proteins or inactivated viruses, etc. And my second question, and I'll leave you with that, is you have such an encouraging product, but it's quite rather a complex product. Do you think that your solution could one day fit with the expectation of the World Health Organization, which is $3 per dose for the country in development? So could you give us a global, let's say, update on the COVID-19 program of CureVac, please, Patrick?
Yeah, I will try to do this in the most appropriate way that I can do this, that I'm allowed to. It's sometimes a little bit picky if you are in a publicly listed company. But it's clear that we are currently in clinical trials with our vaccine. We principally conducted a phase 1 trial and are now in phase 2 trials, also in some efficacy trials that are running in Middle and Southern America, where you have a lot of COVID-19 circulating that you require in order to show efficacy of your vaccine. That you can raise specific antibodies is not enough for getting this to a product level. What you also have to see is that compared to unusual development, this is all accelerated. So the upscaling and all of this stuff also distribution and storage that will be in pandemic vaccine. You cannot compare this to other developed vaccines that are distributed via cold chain, for example, and stored for over 12 or 24 months. Such data is all currently evolving and is produced. Yeah, and what I've seen in the published data, for example, also from our competitors like Moderna or BioNTech, I'm quite sure that we will have an effective vaccine by next year that will be available on the market from several providers. And this will clearly change the way how vaccines are developed and introduced into the market in the future. And I really think that these nanoparticle-based messenger RNA vaccines will be commercial by next year and will change also the perception of nanoparticular medicines. Because you have to see, for example, on Patro, as I already said today, this is for a special people that suffer from this condition. I think a few people. Here we are talking about commercial prophylactic vaccines that goes to millions, up to billions of people, and all of the countries are currently ordering millions of doses. So this will change perception of nanoparticular medicine, I'm sure.
And do you see the $3 per dose an accessible target one day for CureVac, or I don't know if you can comment on that?
I think a concept like messenger RNA can deliver to such goals. It's really about scaling, yeah. And what you also have to consider is that the investment into production that you do is not for only one single product. Yeah? You can produce from the same facility later on also in flu vaccine or in rabies vaccine. So the investment costs are distributed in, over several different products. But here we are talking of a new technology that's just entering, yeah, just entering. And you also have to compare, if you look how expensive antibody treatments were at the beginning of the 2000s and where they are now. There was a significant drop, principally with the increase in knowledge and also production capacities and efficiencies. And I think we will also see this for messenger RNA. So I believe in the end, such goals could be achievable, but not within the first year of this technology in the market.
Okay, well, that was fascinating. Patrick, I would like to first to congratulate for the excellent quality of the talk indeed, but also to thank you personally, because you've been extremely faithful to your first commitment when you say yes for being a keynote in Heraklion in June, and then in September, and then in October, and then for the virtual event, despite the fact that you're extremely busy with all your programs indeed. So on behalf of the whole ETPN 2020 organization committee, thank you so much for this. And we were not disappointed. That was an excellent keynote. Thanks. I'm sure that everyone at home is applauding right now. And thanks a lot, Patrick.
Yeah, maybe as a last word, I also want to thank the ETPN. And I think your contribution to the field is also very necessary. We need such tools. We need the support on translation with all these analytical methods and production stuff. So I'm really also grateful that I had the opportunity to speak here. And I hope that we will be in contact and that you will get the funding also further on, because it's a really important corner piece of nanomedicine technology.
Thanks a lot for these very encouraging comments, and we try hard at least, and you will be. I mean, ETPN is your house, CureVac indeed. Thanks again, Patrick.
Thanks.
So now we come to the end of this first morning session. I hope you've enjoyed as much as we did.